What's up guys, Derek, moreplace.com. Today we are going to be talking about the supplement industry, quality control, lab testing, third-party testing, like kind of what goes into start to finish supply chain to manufacture to actually, finish product with the brand that is selling a product, what goes into that from start to finish. And I have some really high credentialed individuals that are joining us today. First is a senior chemist who has become a chief scientific officer and is overseeing quality control for massive supplement companies in the sports supplement space. Some of them NSF approved, some of them, WADA compliant, this, that. He's done it all, and he's a subject matter expert in FDA quality systems and analytical testing of nutraceuticals and dietary supplements. We're also gonna have on that podcast or, clip, it's not even a clip, it's like a fucking hour-long thing, but we also have David on the call, who is the new COO for Guerrilla Mind. And then afterwards, we have the Guerrilla Chemist. So if you don't know, the Guerrilla Chemist is Brian Moscow. He is an organic and biochemist who has a master's degree from the Georgia Institute of Technology and Chemistry and has been a formulator for a bunch of different companies before, and now has his own supplement company as well, which is on the cutting edge of unique formulations, scientific insight, and is somebody that I think is at the cutting edge in this industry right now, and is somebody that I respect as far as their education, background, willingness to really dive deep into new literature and whatnot, and actually try to, bring out innovations in the industry and whatnot. And they both, the senior chemist I mentioned, as well as the Guerrilla Chemist, we are discussing all that goes into like analyzing products, third-party testing, etc., and getting their interpretation, because it's something that's not my wheelhouse. It's something they have been thoroughly educated in, and I wanted to get subject matter experts on the line to talk about it. And the reason we're making this too is because there has been a lot of talk in the past week about underdose supplements or totally bunk supplements, and the product that has the most attention drawn to it right now for lab testing is Turkesterone. There's a supplement company called Nootropics Depot, and they have access to lab equipment, and what they do apparently is go into their lab and then they test other products from other companies and whatnot. So they have apparently gotten Turkesterone and tons of other products. Like, they've done this for years at this point, and they hold themselves to a supposedly higher standard than the average supplement company. They do their own lab testing in-house. They show all of their QC. They have like these lab reports that show intricate breakdowns of the chromatograms and whatnot of their products. And then they'll show chromatograms of other people's products and show where there may be shortcomings and deficiencies, lack of potency, etc. And they've apparently taken multiple companies' Turkesterone, and they have put them into their lab equipment and have produced documentation to show what they found. And again, we're taking with at face value that this actually occurred. I'm not going to, personally, from a taking this seriously aspect, I'm just assuming it was done, and that's fine for me. We are going to be assessing it accordingly, and that's what the whole point of this video is, and getting into the quality control metrics and whatnot that we're going to discuss later with the chief science officer as well as Brian, and changes accordingly and whatnot based on that, and how to bulletproof testing, essentially, like what is the actual best way to go about ensuring quality control. So anyways, regardless, this guy apparently tests all these products. He found essentially most of them are bunk. Like, most Turkesterone products in the industry are not containing any Turkesterone at all. They are completely absent of it. But then he tested Guerrilla Mind's product apparently, and his finding on his documentation was a significant underdosed amount of the ingredient. And then he found wasn't a cross detection, but he found another spike in beta-ecdysterone. So it's his belief that the product is spiked with ecdysterone and has a much more underdosed amount of Turkesterone. His assertion is that the entire industry is pretty, bunk, essentially, and is, it's a fraudulent industry. And this is not just across, Turkesterone products, although this was like the hyper-focused, product, because everyone wants to see it get crucified, and understandably so, which I'll get to shortly. But he basically showed that his opinion is that, you know, almost every company essentially is failing in quality control, and his lab finds deficiencies often and publicizes them for people to see. And the finding on the turkesterone is basically that every company is bunk. And of the ones that have actual turkesterone, it's really underdosed. So I didn't want to make a video until I had subject matter experts ready who could actually, you know, dissect this stuff with me on a call, and I could actually break it down for you guys and show you guys what has gone into. I can't speak for other companies, obviously, but from our side, you know, what we've done due diligence wise, and kind of break down our lab test results and whatnot. And I just kind of want to address as a preface before we actually get into the real content of the video that there is no situation, like we were an already successful company. Obviously, I value trust factor so fucking much. It would be absolutely nonsensical for this to ever be, if this was actually the result, for it to be an intentional thing. And it's, I think the best way to elucidate that too is to really show the timeline of Guerrilla Mind's turkesterone product, because when we first released it, as far as I know, we were like the only company in the industry that had a solo turkesterone product. Now there were some other ones I believe that had it, but I think we were the only like company that really, I don't know, was well known essentially, that picked up a lot of steam. And it was an unexpected hype train, to be honest. Like I put out a video, just, you know, speculative video on its mechanism of action. You know, I saw that it was being looked, steroids were being looked at by WADA for their ergogenic properties. It had seemingly no HPTA suppression anecdotally. It was like hair safe, like all this shit that seems super promising to me. And I was going to use it because I don't blast anabolic androgenic steroids anymore. So I'm always looking for, you know, other alternatives and unique compounds that can use unique vectors for mechanism of action. Like again, like an ER beta agonist, as well as some of the unique properties that it may otherwise have. And again, some of these are like still to be elucidated though. These are not like conclusive statements. I'm just saying like why I was interested in it to begin with. And you can see that in my initial videos. And like, to be honest, as far as like if it's placebo and whatnot, like I actually understand why why this is such a big deal too, and why people want to see it get crucified. And I totally get it. Like so many companies that clearly didn't really care about the product to begin with, got in simply to sell it based on a hype train. And, you know, that's, I guess, you know, good business principles, I guess, but it's pretty obvious when somebody actually was uniquely interested in the product before there was any hype behind it. Like when I put it out, there was no hype whatsoever. No one even knew what the fuck it was. I barely knew what the fuck it was at the time. I just had a bunch of anecdotes from biohacking bodybuilders and stuff who I knew were injecting steroids. I was like, damn, like, you know, what is the utility of this? Why? And like, why are you injecting it? And it was like to circumvent potential bioavailability issues from intestinal transit and whatnot. It's not very the water solubility is not ideal for being orally bioavailable in humans. And there was a lot of literature to support that creating inclusion complexes with cyclodextrins in conjunction with these, you know, ecdysterone is the main one that is elucidated in the literature, but also turkesterone by extension, I found to be something that was very promising, is something that looked like it had a utility. Like the data looked really promising. The cyclodextrin complex data, I was, you know, really interested in myself. I was like, damn, can we get like an orally bioavailable version of this stuff? Is it going to be like the best format to deliver it? Blah, blah, blah. And I think the best way I can describe like why there is no fucking way I would ever compromise my integrity for short-term gain on a product is the fact that we were the only company selling it for like 9 months or something. And during that time frame, we had big spans of time where we were out of stock. We'd sell it in like 2 seconds. Like that's a bit of an exaggeration, but 2 minutes. Like if you guys were, you know, following since then, you know how quickly the shit sold out. We'd have these like tiny little releases because the material, when we sent it to get complex, we could only do so much at a time. And it wasn't like lack of supply of Adjuga turkestanica. Like we could have literally put raw Adjuga turkestanica in a capsule and sold a fuck ton of it. Once the hype train started at the start of 2021, we were like, no holds barred. We could have had free reign in the market for like 9 months. And instead, what I did was I thought this complexation was the way to go in terms of making like the best version of it. So I sacrificed the obvious short-term game that could have been, if I actually thought this product was a placebo, like I would have just straight up put Adjuga turkestanica in a capsule, sold, you know, bottles of that. Like there was no stopping us from doing that. We could have easily done that, and I could have just ignored all this bioavailability cyclodextrin data that I, you know, was fully aware of, and just been like, look at this fucking rat study showing it compared to D-ball. That's definitely what's gonna happen in humans too. Like this is why I'm using it. Now again, that data was fucking cool. Like, let's be honest. Like that was one of the reasons why I started using it myself. But at the end of the day, like that data could have been heavily leveraged for me to just sell raw turkesterone, or I could have just put like, the cyclodextrin in a capsule and mixed it in a vat like, citrulline malate or something, and then shoved it in a capsule without it actually have the inclusion complex created. So for us, we would come out with these little tiny releases because the actual material needed to be complex. So we had all of this opportunity to capitalize and literally sell 100 times the units. Like, that is not an exaggeration. And anybody who thinks that we were intentionally limiting inventory in order to drive up demand, like, that's fucking nonsense, dude. Like, we would sell it in 2 minutes and then be out of stock for like 1 to 2 months. Like, that's, at that point, it would be nonsensical to be like, oh, like, clearly driving up demand. Like, the demand was so fucking high. We could have sold 100 times the amount and still not have met the demand at the time. Like, that's how insane it was. So the material was always there to sell. And that's why so many companies, once they saw the hype train and wanted to capitalize on it, they jumped on board. And now like a ton of companies sell turkesterone. And that's, expected outcome, to be honest. But I just wanted to hopefully adequately elucidate that, because there was a ton of opportunity cost that I completely walked over on purpose to put out what I thought to be the best version of this product. So if I actually thought it was placebo or bunk this entire time, that would have made no sense. I would have just capped literally the material raw and just been like fucking blasted as many units, and literally had like, I don't know, like millions and millions of dollars more at this point above and beyond any of the sales we had of our product relative to what we could have had. It was literally a disparity of like 100x. Like, that's how insane the demand was for just like, give us anything as turkesterone. Like, it was fucking nuts. So again, we never did that. I never did that. You guys know that, who've been following from day one. I ate millions of dollars of opportunity cost by staying out of stock while other companies, sprinted to get out their competing product. And, I hope anyone who's skeptical really realizes how nonsensical it would be to risk an already successful brand reputation on something that we truly thought was placebo or bunk or anything like that. So our third-party test results show that our product is exactly what it's supposed to be. And that's what we have been under the impression of the whole time. And I feel like delve into that in very adequate detail in the rest of the video. So hopefully you guys, stick around and watch that. But basically, and one thing I did want to preface to you before we get into that is the senior chemist who we talk with, who is a chief scientific officer, has all these credentials. He is not a public figure. He's not an influencer. So he requested that we keep him anonymous. So I respected that. He has been, blanked off the screen, but you can hear his audio, and it's changed, so you can't recognize who he is. But that is a very, very credentialed individual, and you can tell by what he's talking about, he knows his shit. And then obviously, Guerrilla Chemist, credentialed individual, highly respected in this industry, and he's fine with being on camera because he's not like an influencer, but he's like a public, facing guy. So he has his own company, stuff like that. So anyways, I didn't want to put out a video without all the information, having, subject matter experts on the line that I could actually have show this in with accuracy and adequate detail without me trying to just summarize like a week of fucking comments. I just want to cap this off with, before we actually get into the real contents of the video, is the thought of my intentions being like unethical intentions or intentionally doing something to fraud the consumer or something. Fucking painful to read some individuals who trusted me flip their opinions just based on, not hearing my side or not seeing my comments or whatever it is. And it's been a rough week, dude. Like, very, very difficult dealing with this. I have never had like a, like a PR, like big, like blow up thing or anything like that. And for my integrity to be questioned on this has been insanely stressful, especially given that I thought it was very clear, given our position in the industry, even before the turkesterone thing, it would be nonsensical for me to put it on the line intentionally with something that I really thought was placebo or bunk or underdosed or anything like that. It would make no sense. I appreciate you guys, watching through this in its entirety and listening. For those who want to watch are probably the only ones tuned in at this point, because I understand it's kind of boring discussion, but this is something that like I'm taking very fucking seriously. And if we identify holes in quality control or anything like that, like, they will be addressed accordingly. And I'm going to show exactly what we have done to date and why we are under the impression the product is legitimate. But if a certain batch turns out to be otherwise, it'll be addressed. I fucking promise you that. So let's get into the actual content. In establishing a new product and working through the sourcing aspect, what is the qualification process for new ingredients?
Okay, so simply put, what you really have is under 21 CFR Part 111, there is a preface for what's called a vendor qualification program. All right, and in qualifying a new ingredient, the first thing that a group inside a company, nutraceutical company, would do is they're going to have to make a specification, all right? And that specification is going to include a number of requirements. And then to some extent, it's also built upon what is important from quality variables for processing of the final product. So for instance, if you have, let's say, a processing aid that is used if you're making a tablet, then one of your excipients would be what's called a DC grade or a direct compression. And in some instances, an active ingredient can also be bought that way. So in a direct compression, it's going to have a larger particle size, a larger density. It's going to enable flow on a high-speed machine. In other places, when you talk about active ingredients, it has a lot to do with the first and most requirement, which is identity. And the reason I say that is because what the FDA has put into the 111 standard is that you must confirm the identity of every ingredient every time you purchase it. And a different part of the program that I'll get into in a moment, we'll talk about a different phase of it, which is called qualification of the certificate of analysis, or C of A. So what we're really doing when we establish the new product specification and the sourcing of it is you're evaluating both the material and you're evaluating the vendor. And so from the vendor standpoint, you want to make sure that they have a program where they have examined the material and found it to not contain substances such as heavy metals or micro content, especially from a pathogenic standpoint, that would be harmful and be a safety issue for people to consume, all right? In addition to that, it would have to meet whatever claim it has. So in our industry, we look at buying everything from powders to extracts to what we call standardized extracts. And in the powders, you simply have a plant part that contains a substance or a family of substances that have therapeutic value, and it's basically dehydrated, it's dried. And that powder then would have molecules that you would want to compare to a standard using several different methodologies. The reality is that that powder ends up having many, many different molecules in it. All right, because what happens when you move to an extract is you're now going to take that same plant part and you're going to run either like water or alcohol or a combination of which is a hydroalcoholic extraction or with other solvents. And that's going to solubilize and concentrate therapeutic molecules in a greater degree. So now when you do an extract, you're generally going to leave some of the plant parts behind. All right. So we tend to call those ratio extracts. And the ratio extracts depend on how much starting material you have versus how much material you extract at the end. So if I buy a ratio extract and I start with 10 kilos of material and I end up with 1 kilo of material, then when you purchase that, I'm going to call that a 10 to 1. All right, it could be a 5-to-1. In some cases, like with your 95% curcuminoids turmeric, it's usually like a 65-to-1. Okay, so it just depends on that ratio. Now, in that case, I've now talked about what the powder is versus what a ratio extract is, and then there's the standardized extract. And actually, the 95% curcuminoids would be a great example of standardized extract, because now you know that there's a minimum content of curcuminoids in the material that you buy. So you have these different extracts and or powders, extract, different active ingredients. And when you establish your qualification process, there is going to be differences in the specification that you write, because when you buy a powder, it doesn't really have a guarantee of the therapeutic molecules in there per se. So doing an identity test is going to send you in a direction where you're using typically what's called TLC, or thin layer chromatography. A new version of thin layer chromatography, which is a whole lot more friendly to the industry, is called HPTLC, which is high pressure thin layer chromatography. So in powders, you're really looking at that HPTLC or TLC method for identity. When you get to a ratio extract, you're still probably going to go that direction, because again, it doesn't guarantee something. You go to a standardized extract, now you're going to have active molecules in a known minimum quantity. And in this process, then what's going to happen is you're going to move into more accurate just different analytical techniques. Like, for instance, if the active ingredients are a water-soluble molecule, then you're going to use HPLC, which is high performance or high pressure liquid chromatography. If it happens to be a fatty substance, especially when you talk about things like omega-3s and so forth, you're going to use gas chromatography, which is GC. All right. So these methods allow you to both quantify a standardized ingredient, and also every test you run when you're analyzing a material as a comparison of a known versus an unknown. So everything has to have a standard. So when you run these tests, that is going to become like your identity test, because you're going to compare a known standard, which the laboratory will have, versus the material that you're trying to qualify. And so that's the most important process that you're going to do, because it is required every time by the FDA. And the short answer on the reason why is because a lot of these materials are very expensive. And so there is a lot of potential, especially from overseas purchasing, where there's fraud. And so in order to understand that you got good material, high quality material that will have therapeutic value, the identity test is extremely important. There are times when, even in not just the nutraceutical industry, but in the pharmaceutical industry, you have seen where there has been fraud that has been deadly. All right. The first thing that comes to mind to me is heparin. And you had quite a few people end up being killed by a company that found a similar molecule that fooled the USP test for identity of heparin. And what they're doing is they're conducting fraud in an economically motivated way. And so that is the thing that the FDA wants you to absolutely be your most important test. All right. So qualifying a material is going to include identity. After that, we have different categories of things that we look at. And those categories can include physical properties of the raw material, chemical properties of the raw material, and in some cases, what we call organoleptic properties. And organoleptic is a property that your senses can detect, all right? If it happens to be a flavor that you're putting in, then one of the organoleptics would be the taste or the smell. If it's a strawberry flavor or a chocolate flavor, et cetera. Analytically, there's a couple that are safety tests to show that material is safe to consume. The one that stands out the most would be the heavy metals. So typically, arsenic, cadmium, lead, and mercury are the four sort of heavy metal panel tests that you would do to ensure that we don't have levels that would potentially harm somebody. And that could be either in a single serving or servings over a long period of time. Another concern would be the micro content, because so many of our ingredients are plant-based that you're going to have a natural microbial flora that's going to associate with the material. And when I go back and talk about the powders versus the ratio extracts or the standardized extracts, the powders are typically, because they're just dried, they're going to have some micro content to be concerned about. So we want to do everything from TPC, which is total plate count, to total coliforms, and yeast and mold. And those are standard tests where there can be some content, but we don't want the content to be very high. In addition to organisms that don't harm us, but we don't want a high content, then you talk about the ones that would, which are the pathogenic microorganisms. And so we specifically look for a negative test, no content, no presence on there. So basically, I'm covering that in the qualification process, you can build your specification off of identity, off of the actual assay content, when there is a known therapeutic molecule concentration, the heavy metals, the micro, and then physical properties. And to talk briefly about the physical properties, we talk about things like density of a molecule, the size of the particles. And in this case, this has usually more to do with processing in the manufacturing plant. Moisture content can be important. And so these physical properties also want to be put into the specification that you're going to write. And just to make mention here, what you really need is kind of a team of people to write your specifications. That's your best case scenario, because there's going to be analytical parts where you have a person, a representative from the laboratory would come in and contribute from the laboratory side. Then you would have a formulator who's in R&D, product development, or supporting manufacturing, that is going to look at the physical properties to make sure that it will be able to be processed on a high-speed machine. And then you also need someone from your quality unit, because they're going to come in and they'll be the ones that will actually handle and control the documentation aspect, the document control of the specification itself, as well as making sure that there is a collection of the correct tests to qualify the material. So when you put all that group together, you should be able to build a very robust specification that is part of the qualification process, or vendor and ingredient qualification process.
So presumably that document I put in the chat box there, that is the certificate of analysis spec sheet, basically elucidating everything you just described, from appearance identification with TLC, heavy metals testing, microbiological, etc., as well as the actual HPLC test results for the content of the standardized material. So that kind of like checks the boxes, or I just wanted to kind of get an overarching summary, ideally.
Yes. So when you look at this particular certificate of analysis that you have some physical parameters such as the appearance, some organoleptic, in the odor and taste. You have particle size, which is described as the mesh size. The loss on drying is going to look at sort of the moisture content. You have the heavy metals with the lead, arsenic, cadmium, mercury. You also have a potency test here, as well as the biological tests. Yes.
Okay.
So this is a complete document.
Okay, awesome. So then from that point on, like at that point, you can confirm it's met like minimum requirements to be, it is what it is. The assay is, showing the potency is where it should be, at least for the constituent that is standardized from the big, the raw material. It's not contaminated, et cetera, et cetera. So from there, how do you ensure ongoing quality process, like from that point on?
Okay, so essentially that's like the second half. Once you've built the specification, and which typically is going to have the same parameters that are on a certificate of analysis, then the qualification of that material in Part 111 is going to come away with monitoring the testing, especially for the beginning lots that you buy. So you're going to have the test results, and you're going to compare it to your specification, and then your quality unit is going to look at those results, whether the testing is done in-house or is done through third-party analytical labs. And then they're going to make a decision on whether that material is approved for the lot that you purchased. Typically, what the industry has done through coordination and understanding from the FDA is that there's a acceptable minimum number of lots that have to be bought in succession and tested and meet the specifications before you get to a point where you have, at that point, not only approved the material, but you're now qualifying it, and you have an opportunity to do a secondary thing, which is called qualification C of A. So before I talk about that, just briefly, I want to say that when you buy material from a vendor and that material continues to meet the quality variables, then you go through a evaluation of the material, not only the one material that you buy from a vendor, but you're really looking at more than one material if you end up buying more than one material from that vendor. So for instance, if I buy 3 different active ingredients and 1 processing aid from the same vendor, and I evaluate, and let's say the first 3 lots that I buy of each of those ingredients is passing all my specifications, then I can capture an approval of qualifying that vendor as selling me good material. Okay, so that is not only focused on the material, but on the person who is selling me the material. So in that qualification of the vendor, I have a lot of data to prove that this person is truthful, honest, and selling me good material. Okay. In addition to that, vendor qualification usually involves either an audit of their facility and the associated quality system that they have, or it is a audit questionnaire that someone will send so that they better understand For instance, your company would better understand what the company has in place as a quality system, what aspects of the warehouse that they store their material in and how that's controlled, etc. So typically these supplier questionnaires are sent out, and that becomes a necessary part of the vendor qualification as well. So the vendor will then fill out, what the size of their warehouse is, whether it's temperature controlled or not. They'll talk about their quality system. All right. They'll talk about, because they have a place in Part 111 as well. So they have to keep their warehouse clean. They have to have a document control department. They have to have a qualified individual who runs their quality unit. And so they have to have all these things in place as well. And then that's a different element of vendor qualification as well. Now, when the FDA was putting together 111, there were a lot of question and answer sessions held across the country over a couple of years. And during that time, the FDA was communicating and broadcasting what they wanted from testing. And as we know, Part 111 looks very much like sort of a cut down version of Part 210 and 211, which is pharmaceutical. So in this, there was a lot of pushback from the industry about testing. And the FDA's position was, well, we want you to do a lot of testing. We don't feel like the nutritional industry is doing enough testing. In our inspections of facilities, we tend to see that people have a raw material come in, they get a C of A, they take a look at it, they file it away, and then they use the material. And I say, well, that's not good enough. All right. First of all, you have to know that most everything you buy is going to be a white powder or a clear crystal or something like that, or possibly a brown powder for plant material and so forth. And they said, we've got to you have to do identity. So as I explained earlier, that is a must, and you must do that. After that, then they wanted other testing, but the industry pushed back and said, look, we don't have the same financials as the pharmaceutical industry does. So we can't do this whole host of testing on every lot we receive. In addition to that, there was also a lot of good input from the standpoint of, let's say there was a reputable manufacturer who'd been in business for 20 years before 111. And he said, I have a mature relationship with a vitamin C company, and now you tell me I can't trust them and I have to do full testing on every lot coming in. And that person might say, well, I test my finished product and I always have a passing assay. There isn't any issue there. So that's where qualification of the C of A came in. And in the qualification of the C of A, when you buy your material, such as the material in the C of A that you have here, then you do testing against any one of these quality variables, except for identity. Any one of these quality variables that you want to qualify. And then after the qualification process has happened, typically after 3 individual different lots that are purchased, then you may rely upon the C of A to meet your specification. And there's a lot of misunderstanding there. So for the most part, people will select the assay quality variable because it tends to be the most expensive. The microbiological is not very expensive, and the heavy metals generally is not very expensive. So with the potency, once you test lot number 1, you get a pass. Lot number 2, you get a pass. Lot number 3, you get a pass. Then when you purchase the 4th lot, you still have to meet your specification. But instead of sending it out for an assay, because the first 3 assays for the first 3 lots purchased all are within specification, and they more or less, they match the value that you have on the C of A, you may use the value on your C of A to meet your specification. So in the case of the turkesterone here, you have a minimum of 10%. So this is a standardized botanical extract. And so you test your first 3 lots and you get 10.5, 10.2, 10.8, and then the 4th lot comes in. You've already qualified the vendor and you qualified the assay as being truthful and accurate. Then instead of sending that out for assay, you may report the value, in which case this lot says 10.73%. You can use that value to meet your specification and approve the material. Does that make sense?
Yes. So as far as for us, obviously we're not the manufacturer. We private label through manufacturers. And for us, like, we rely a lot on their quality control process, being a GMP certified facility that's, FDA regulated and is audited regularly. But then in addition to that, when we get the finished material, we, as like a company that private labels products as well, sent a random bottle in for our own third-party testing. And this is what we found through our own LCMS testing, separate from manufacturer quality control. And this is, I just linked it in the chat. It's the first LCMS test result.
Okay.
So in this one, basically the way our product's broken down is it's based on data on cyclodextrin ratios for an inclusion complex. And I wouldn't expect anyone to know what that is really, but since day one, I was under the impression, based on a bunch of literature that I saw, that the best way to ensure maximum bioavailability of ecdysteroid products is to complex it with a cyclodextrin. You see a massive spike in water solubility and ability to make it more orally bioavailable in humans is what the data suggests. So that is what we use as our basis for forming what we saw to be the best version of this product, which at the time, I don't even think there was very few solo turkesterone based products out there, if any. I think there were some, but they weren't like that popular. And to be honest, I was kind of very shocked at how popular this even became. I thought it was just an interesting compound that looked like unique. And, WADA was looking at ecdysteroids as something to add to their watch list because it had potential ergogenic effects in athletes and whatnot. It looked really promising to me. So anyways, the breakdown of our product is, if we just put raw Ajuga turkestanica standardized to 10% in the capsule, 500 milligrams of that would yield 50 milligrams of turkesterone. But for us, we have this ratio of hydroxypropyl beta cyclodextrin, as well as the Ajuga turkestanica extract, which is standardized to a certain amount of turkesterone. So for us, this LCMS, we would have been anything above 25 milligrams is like, for us per capsule, what we would have been happy with based on these ratio studies. And that's what we saw in our LCMS. Like, we see the HPBCD, hydroxypropyl beta cyclodextrin, is within like, a handful of percent of 250 milligrams. And then the other part of the ratio would be the Ajuga turkestanica yielding this amount of turkesterone. So based on this, like, I don't know of a higher sensitivity test based on the fact that the material itself is HPLC'd before getting sent to us, the quality control of the manufacturer, and then for us, we're mass specking it with liquid chromatography too, to ensure it is what it is without the manufacturer even knowing. So, like, your interpretation of this test result, is that like sufficient and valid, or what would your interpretation be?
Yes, this looks sufficient and valid because you're setting a specification yourself for minimums, okay? And in doing this, you have not only engineered the combination here for a high bioavailability, but you've also put this into a high level of testing, okay? Because when I saw the turkesterone on the raw material C of A, you see that it's HPLC, all right? So you're taking what is a molecule that can be seen on an HPLC as a raw material, and then you're taking it to a higher testing standard by using LCMS when you put it into your final product.
Mhm.
So you have in this result something that is uh, a healthy level above your minimum specification.
Yeah.
So this does qualify your material as meeting the design and the label claim.
Okay. Awesome. That's good to hear. See, that was a huge relief, hearing that off the bat, because obviously, some of the rumors circulating, it's kind of like iffy stratifying for this ecdysteroid because there's so many similar molecules and stuff that it seems like there's a lot of chemistry debate as to even figuring out if it's there, if it's cross detecting as something else, what is going on. And it seems like the chemistry, whoever is interpreting the data, depending if they're biased or not, like I can't think of a higher level than an unbiased third party ISO accredited lab that has a good reputation among those who like big manufacturers in the industry and companies. And like from what it sounded like, ABC testing, has come under a little bit of scrutiny because of the opinions of some on this FDA letter they received. I don't know if you're able to speak on that in any capacity about how it's based on the pharmaceutical side versus the dietary supplement side. But from what I've seen, they've always been compliant on the supplement side and they're held in high regard by a lot of people. And as far as I know, pharmaceutical companies receive warning letters all the time, like Pfizer, Eli Lilly, blah, blah, blah. Like, I feel like that's pretty common for pharma companies on a regular basis, like even the ones that are held to the most stringent of standards. So it's like separating supplements from pharma, I feel like is important when people are drawing conclusions about like the actual credibility of a company. So like presumably they've retained their ISO accreditation, are still used by big companies. Like you believe them to be like a good source for third party testing, presumably, or what's your opinion on the situation?
Well, yes, I've used ABC testing in the past across several companies that I've worked for, whether they were a small company, a medium company, or a global company. And the ABC testing laboratory was created by a person who was originally a professor of botanicals and had this great knowledge of the analytics that are associated with them. And along the way, the lab grew in reputation and really what is the hardest industry to test for, which is nutraceuticals. You mentioned the pharmaceutical industry, and they do testing for the pharmaceutical industry. They do testing for a medical device industry, as well as nutraceuticals and foods. And yet, it takes a lot of expertise to jump, across those paths. Specific to my knowledge of the FDA warning letter, it had something to do within the medical device industry, and it was something that was taken care of, resolved, and so forth. There has been no activity or warning letters or anything relative to nutraceutical industry. When you look at analytics in the nutraceutical industry versus, say, for instance, the pharma industry, the funny thing is as a chemist, you have to be a lot better trained than you do in pharmaceutical industry. And let me explain why. When you make pharmaceutical, you typically have one ingredient that's active, all right? It's salicylic acid, it's diphenhydramine hydrochloride. It's not even in a large concentration, okay? The rest of the tablet or capsule or soft gel is processing aids. So you don't really need much of a chromatographic method because you're not really separating anything. Okay, you have one active ingredient. So when you talk about the nutraceutical industry, it's the difference between a tree versus a forest.
Yeah.
And so your separation techniques have to be so much better. Your understanding of the methodology and so forth has to be so much better. And here you have someone who's an expert in botanicals who creates a lab and has these analytical tests where the chromatography is built to a much, much higher level in the pharmaceutical industry so that they can get through all the biological molecules that are in a powder or an extract to find and see and separate the molecule of interest. So it's sort of ironic that the pharmaceutical industry sort of looks down on nutraceutical when in reality our analytical methods are far superior.
Yeah. So circling back, like it's good, obviously reassuring to hear on ABC because we've done quite a few tests with them, especially on this product. So we had a second LCMS test result, and over the time, like obviously this material is always, it doesn't even make it to the manufacturer without the identity, the HPLC analysis showing the active material from the standardized, the raw material at the start. And then they have their quality control at the manufacturing level. And then we have this other third-party test as well that shows it's the second to the last link there. And it shows an LCMS result of our complex of cyclodextrin to adjuga turkestanica, and then a corroborating report with the raw material being assessed via UV vis hitting over 10% spec. So these are for the same like month time frame of the same raw material batch. One of them is the capsule itself of a finished product that was randomly selected, and then the other one is the actual raw material itself. So I was wondering if you could touch on the results of this a little bit, like at the high level, we have the HPLC, we have quality control at the manufacturer level, and then here we have this LCMS again, but it's for the actual complex itself, and also this UV vis with the turkesterone resulting in a 10.05% result.
Okay, let me load this and have a review of it. Okay, so in this case, you have a test of the product at a later date.
Yeah, so this is in March of this year, so corresponding to the most recent batch.
Okay. Yes, and here you absolutely have still a passing result, meeting spec as well from the UV vis standpoint. This, again, is above and beyond what is required as a minimum, okay? It is important for people over time to test their product to make sure that if for some reason there's a stability issue, that you can make adjustments to your formula, you can make adjustments to your packaging components, for instance, adding desiccants, things like that. And even putting on your label things about storage conditions to keep the product as fresh and as stable as possible. So I'm glad to see that you're doing this, because this kind of separates you from other people in the industry.
Yeah, like I'd be shocked if any other, like most companies I feel like aren't even sending anything in at all. It's just like relying on the COA entirely, and then that's kind of it. Like, is that kind of what you see across the industry, or what's commonplace?
Well, I think that, what you end up having is that when you're using companies who have a third-party GMP certification to them, then you as the brand owner have separated, the people who are more economically motivated in the industry versus the people who are more focused on quality and long-term sustainability as a co-manufacturer. What they're doing is participating in these third-party GMP certification programs. They're audited, depending on the program, once or twice a year, where they do a complete evaluation of both the company's quality system and the physical property. And they basically go through the entirety of Part 111 to evaluate the compliance. And so most of them will do sort of a grading standard, but depending on who it is, it can be a couple different ways they evaluate them, and this is an ongoing thing year after year after year with these people. So you know that these people are doing the tests that are required. You know that these manufacturers are qualifying their vendors and are buying good material and making product that meets label claim.
Right. Okay. So based on the, all the test results you've seen and kind of just like understanding how quality control works in supply chain from top to actual seller, like presumably we've done the due diligence that you can perceive. I just want to make sure like we're all on the same page here.
Yes, I believe so. So the idea is that you have a vendor buying raw materials that meet quality standards. That vendor is then selling it to a manufacturer who's evaluating it for quality standards. And then you, as the brand owner who received the finished product, you are turning around and evaluating it for the same quality standards. So
Yeah.
the idea of the quality system there is that if there's ever an issue, whether it be accident or by intention, all right, is all three facets of the supply chain from upstream to downstream are using verified techniques, assay, spending money to confirm the quality and adherence to the label claim. And on top of that, if there's a problem, you're going to catch it before it ever gets to the consumer, which is one of the most important things that the quality control unit in any of the three facets of the supply chain is, that they catch a problem before it leaves the facility.
Okay. And I guess one thing I did want to touch on is some of the chemistry when it comes to variability between the chemists who are interpreting the data or however it may come across. Like it seems like, David, you gave me an example of KSM and Sensoril ashwagandha companies have produced their own with analytes test for their own ashwagandha with analytes. Let's see, like it's not a standard US standard methodology and test. If using the US standard for with analytes testing, the test would actually show lower levels of the ingredient than the two branded test methodologies will show in their specifications. They develop their own versions to show their withanolide levels and let everyone know. Can you verify a finished goods makeup with your own test methodology like a KSM or Sensoril does with their own withanolides testing methods, and what can cause issues? So like there's definitely variability in terms of interpreting ecdysteroid data, especially because these are like novel compounds without much data surrounding them, or even like, how to go about doing it essentially. So like even at the level of ashwagandha, which is like a very, very popular ingredient used widespread across the globe and has patented ingredients designed around it. What do you see in regards to these like testing methodologies? Hopefully that wasn't too long-winded. I apologize for the length of that.
No, I think, you're giving a really comprehensive scope of variation in test methods.
Mm-hmm.
So generally, when you're talking about a test method, you have a couple different parts of it, okay? You have extraction of the molecule of interest into a solution, all right? Then you have settings in the instrument that will allow for chromatographic separation. Then on top of that, you have to control the detector. And so generally speaking, when you talk about setting the detector, where you're looking at anything from the UV vis, which is the ultraviolet and visible spectrum of light, to infrared, such as in FTIR or NIR, being near infrared light. So you have to set it to certain wavelengths, and these wavelengths are the wavelengths where the molecule of interest has an absorption. All right. So typically, there isn't a lot of variability in these methods when it comes to setting detector, because the molecule of interest has documented what is the highest absorption wavelength. It could be 230 nanometers. It could be 380 nanometers. What it comes to in the variation here tends to be the chromatographic separation. So chromatographic separation, for instance, involves when you talk about liquid chromatography, you're talking about the liquids used, and then also there's a separatory column. All right, so different methods can use different columns. They're usually going to have very similar properties, but not exactly the same. And oftentimes you can use a slightly different combination of liquids to get separation. But you have to remember again here that when you're talking about the nutraceutical industry, you have a lot of things to look at in the finished products. For the raw materials, it's certainly a lot easier, but it doesn't have to be the exact same method, because the reality is that typically a molecule is soluble in more than one type of solvent. Okay, and so if a person uses a methanol and acetonitrile combination, one method might have a 50-50 blend, another might have a 25-75 blend, but you're still getting solubility of the molecule. And so it's a little bit of an art at that point. So if you have two people trying to paint, a vase, their art isn't going to look exactly the same. All right.
Right.
But the reality is that you're still seeing a clear and visible quantitative amount using different methods.
Okay. So, like, for example, our recent ashwagandha, the most recent batch of it that we've used in one of our products, I'll post a chat link. So this is a HPLC result for the withanolide glycosides content for a generic ashwagandha. So obviously for like variability's sake, I feel like it might be worthwhile to go to like a KSM 66 in the future, just so I don't have to, worry necessarily at all. But it seems like this generic definitely hits spec too, if I'm not mistaken. And this is the assay you would be using to assess that, presumably. Like there's no, I don't know, like HPLC for this type of constituent is what you're standardizing for is what is it, valid and acceptable, presumably, right?
Right. And so typically what you're going to see is, and first of all, notice that as you've gone across different botanicals and unique compounds and stuff, you're able to use the same laboratory. Mm-hmm. Okay, that's one of the things that the reason why ABC is so good at this is because not only do they have such a wide array of methods and analytical techniques and the expertise to do the proper separation so that they can get these results to be accurate and truthful. Try calling other laboratories around the country and see if they have as many botanical analytical methods for quantitation.
Yeah, that was part of the reason we ended up there for turkesterone testing, is because, like, especially when we first put it out, there wasn't very many companies selling it. So it's not like it was a commonly asked for test. And getting a standard, for example, from Chromadex or Sigma Aldrich, like very, very expensive. It's like a unique thing not a lot of people knew about, and they were one of the only ones who was capable of doing it. So, like, I feel like that speaks for their, ability to produce like high quality chemistry, presumably.
Absolutely, it does. And that's exactly the point I'm trying to make, is that there are oftentimes you don't have many choices. And in this case, you have a laboratory that has that expertise across a wide number of molecules, whether they're botanical oriented, they're synthetic oriented, et cetera. So they definitely have a great deal of expertise when it comes to analytical and chromatographic separation, so that they see the molecule of interest and no interference when it goes through the detector. But what you have here is you have the HPLC method. but you don't always get the details on what are the solvents that you use. Did you have to heat the column? What's the temperature of the column? What is, the speed of the pump? What is the amount of the injection? What kind of column did you use? What wavelength did you put it to, and so forth. So you get HPLC, you get the analytical instrument, and that's what you get. So it is hard at times to figure out expertise across multiple laboratories, and so I think that it speaks a lot that you're willing to go through different laboratories. Like, for instance, something I used to do was to take the same ingredient and send it to different laboratories and say, well, what are they going to get? What's the variability in it? And if you have one that comes back and says it fails, and the other 2 or 3 laboratories all say it passes, then right away some of that one laboratory where it failed, they did something wrong. And so it's a good thing to say here that when I've talked about vendor qualification and a compliant quality system for Part 111, not only does it talk about the vendor, the ingredients, it also is laboratory qualification, right? So there are times where I have sent a failing sample to a lab on purpose just to make sure that they're doing the test. They're not what we call a dry lab, where they get your sample, they report a passing value, they never ran the test, and so forth. And as a lot of people know in the industry, that there was a particular laboratory that was caught doing that on camera. So these things are real, and so again, I feel like you're using a very trustworthy laboratory that handles an immense amount of work that companies all across the United States are using this laboratory because they know that they have the expertise and so forth. Is very important, but it's also qualification of the laboratory, and so they have to have certain systems in place, and so there's not only good manufacturing practices or GMPs, but there's also GLP, which is good laboratory practice. And typically, one of the most recognized evaluation of a laboratory is if they're ISO 17025 certified, all right, because that is a laboratory accreditation, which ABC holds above many other accreditations. So, but again, when you talk about the actual test here, you're coming back and you're getting an analysis that is passing the specification. Where the reality is that the KSM material is probably the best in the industry. I am not an expert on that, but they've been in the industry for a long time, not only for supplying ashwagandha, but KSM is very active in the trade associations and so forth. They're extremely active, what we call branded ingredient. Okay, so branded ingredients not only use extracts or molecules that are tested really to the nth degree, but they generally have a lot of clinical work showing that they're therapeutic as well. Branded ingredients cost more money, so the people who buy branded ingredients want the highest quality there is. They want something supported by clinical studies and so forth. So in the fact that you're considering the KSM and other branded ashwagandha ingredients, again, separates you from the guy who's has ashwagandha in a supplement and they're not doing the level of testing you're doing. They're probably at a lower price point, and so people buy a supplement that is an unusually low price. You're probably not getting what's on the label. That's right. What it comes down to. So this is what you can do. This is the effort is you're spending the extra money to make sure that you have the data to the best of your ability, because so many branded ingredient owners, you don't have full-time chemists or laboratories doing things. You have to rely on third-party laboratories. This
Yeah.
is this is not a branded ingredient person's expertise overall. I mean, you speak very well for all of the test knowledge here and the molecules of interest that you're putting into the supplements and so forth. A lot of people don't have all that knowledge.
Yeah. When it comes to patented ingredients, before I get off that topic, so I know KSM 66, Sensorol, like these are ingredients that have, tried and true testing and have been, backed by clinical literature, et cetera. For some other ingredients, like, what would you think if you had a patented material that tested at spec at a bunch of labs, but then another lab found it was underdosed randomly? Like, is that, just like chemistry, kind of going back to the art of chemistry? Is that kind of just interpretation of that? Is that like a result you would take seriously, or what is your interpretation of that?
Well, I think, first of all, it would be very questionable on its own. But to actually be in a situation where it was sent to multiple laboratories and one reported a low concentration or no concentration versus other laboratories being able to easily find it and quantify it, and their quantification match the input that the manufacturer used. That would say that there's probably something wrong with either the extraction part of that particular laboratory, which would also suggest that they have a lack of expertise. And so, again, that's a good reason to disqualify a laboratory, is when you have something in there and you've seen it in laboratories that you've already qualified, and then you send it to a new laboratory, and or you hear of a result coming from a laboratory saying, well, your product was pulled off the shelf and we didn't find anything. And here you're spending all this money to show that it is there. Then there's something else going on. Either it's a bad analytical technique, or it is some other, let's say, economically motivated reason for that kind of thing coming out.
Is it possible that a product or an ingredient that is standardized to some specific amount of whatever the target is could still get granted a patent, though, if they weren't as rigorous with multiple labs? Like, is it possible to just test it at one lab, get the patent granted, and it actually does fail at multiple labs elsewhere? Like, I'm not sure the exact process that goes into approval of one of these like patented ashwagandhas or some other ingredient.
It would be highly unlikely, is the reality. I mean, in certain cases, sometimes the technology of how something is made requires a unique extraction method. For instance, I can remember years ago when beadlet technology was used for beta carotene. That was the beginning of when you had to use enzymes, because in beadlet technology they would spray dry on like a sugar sphere a layer of beta carotene and then a coating, and then another layer of beta carotene and a coating on the sphere, and they would build up the beadlet. And if you didn't use an enzyme to dissolve all the coating layers, then you would only extract the top layer of the beta carotene, and then you get this really low result. So if a laboratory is not using the enzyme technology to go through the other layers of coating to get the full amount of beta carotene, you're going to come up with a low result.
Right. Okay, that makes sense. One thing that is interesting about this Adjuga trichestinica plant is the constituents of it. There are multiple turkesterones. Like, there's a bunch of different ones that make up the whole plant. Like, when you break it down to this, multiple different extracts, you could standardize it to, I guess. Like, it has ecdysterone in it. It has a whole bunch of other things. So is it possible to have at what level of specificity can you conclude there's no overlap of these things, because again, like this is such a new unexplored area of, I don't know, like dietary supplement, that to me it doesn't seem unreasonable that sometimes you might have a cross detection of, like, I can understand at a UV vis level why you might cross detect like ecdysterone as turkesterone or something like that. But then at a higher level of HPLC, is that still possible to do? And then when you add mass spec in there too, is that possible to have happen if they're like very chemically similar? Or is that impossible if you have a standard from Chromadex or something and a good chemist?
Well, and yeah, I think what you're describing here, first of all, when you talk about UV vis, you're probably talking about performing an extraction where you have um, the molecule of interest, or in this case, you're talking really kind of about a family of molecules, okay, which is very common. You're going to have in when you talk about plant extracts and therapeutic ingredients in plants, sometimes it's not one specific molecule, it's a whole family. Uh, and so when you do the extraction, you remember I talked earlier about the fact that you always have a known versus an unknown. Okay, so if the known is a standard is only one molecule from the family, then you're not necessarily going to see the rest of the family, right? So the standard is oftentimes has to have multiple compounds in it. Catechins is a good example. There's quite a few different catechins that make up the entirety of that family of molecules. So, but not only that, then what happens is when you talk about UV vis, you're really talking about a spectrophotometer. Spectrophotometer is basically an instrument that is just the detector. Okay, so you don't have in liquid chromatography, you manipulate your solvents in a way to separate molecules. Well, you don't have that in a spectrophotometer. You just have the detector. And HPLC, in addition to different solvents that you can manipulate for physical separation, you also have a column for physical separation. You don't have that in a spectrophotometer. So UV vis is really just, making an extraction and then putting it in the detector and you get a reading. So if it's a raw material, you don't have interference, but if it's finished product, you do. You have other stuff in there. So there's a high possibility that you're seeing other molecules that are going to have absorbance across a wavelength of light or a spectrum of light that includes a wavelength of interest for the molecule. And so you're going to get a higher reading. It's going to look like it's more potent than it really is. When you get into HPLC, now you have that liquid manipulation to separate compounds, and then you have the column, which further separates compounds. And then when it goes through your detector, you're going to get more accurate reading. And so in a family, it also then depends on, the known is your standard going through that detector, and then the unknown coming through. If the unknown is more complex than the standard is, then you may miss some of that and you don't get that. When you go from the spectrophotometer using just your UV vis detector to HPLC, where you have now the liquid and the column making chromatographic separation, then by going to LCMS, you now have a mass spec detector there that is going to give you further quality information on the mass of the compound of interest. And so, you're upgrading every time you do that. You're going from, your UV vis spectrophotometer is the back of the plane, and going to HPLC is a major upgrade. Going to LCMS, you're going to first class.
Okay. That's a good analogy. That makes it easy to follow. Okay, so like presumably, if you have your UV vis, you have your HPLC results, which at the start of the supply chain, or at least one layer under, we have identity being established, as well as the standardized material whatever concentration and potency is assessed using HPLC, which is the liquid chromatography step. And then you have quality control at the manufacturer level. And then above and beyond that, if you're one of the people who is actually going to randomly assess the quality control of your manufacturer by pulling a raw bottle and like sending it in for testing, and you do an LCMS, like there's nothing more you could really do above and beyond that. And if it shows it clears and it's passed, like you have what you should have, presumably. Like there's no.
Yeah, you don't have really many more options than that. You're going to the nth degree here. Again, you're covering the supply chain from upstream and downstream. You're selecting labs that are passing the qualification process. You're using the raw materials that pass the qualification process. And then on top of that, as a brand owner, you're doing further testing just to make sure that there isn't something else that happened along the way, and you're getting passing results to label claim and specifications. That's pretty much what you can do.
Right. I think, David, do you have anything to add? I think I'm pretty good as far as, had my questions elaborately answered, and I feel a lot better about understanding this whole process and ensuring everything's dialed in.
Not right now. I think that you really covered all the points clearly.
Okay, awesome. All right, I really appreciate you taking the time to get on this call. Like, obviously, this is something that is hard to wrap your head around if you are hearing about it for the first time. I don't think a lot of people understand how the supply chain works from start all the way to finished product. And just hearing, it elucidated from somebody in such a accredited, high level position as yourself, it's very reassuring, also very insightful. And I think a lot of people probably learned a lot from today. I really appreciate your time.
Well, thank you for the opportunity. This is something I love. It's a life work, and I find that there's quite a few professionals in the industry who they love what they do. And it's important. We're making therapeutic products for people, and we believe in plant medicine. We believe in these other compounds that work. So it's my pleasure.
Awesome. All right, so we're here with the Guerrilla Chemist, and I am pleased to be able to bring you an actual, chemist to the discussion and kind of elucidate some of these, the intricacies when it comes to analyzing supplementation and whatnot. And we will be doing a longer form podcast very soon as well. And I don't know, to discuss, novel ingredients, also your fertility protocol recently, something I've been, going back and forth with you. And congrats, by the way, for getting those numbers up. That was really great to see.
Appreciate that.
Yeah. And as far as, okay, so there's been a lot of talk over the past week about like quality control tests, how to assess for things that have cross detection potential, stratifying steroids from like the parent material, this and that. Like, could you speak to just general quality control metrics from like like a supply chain aspect for what you expect from raw material side of things, getting down to a manufacturer, what GMP certification kind of like means in general in terms of like what they're doing at that level to ensure the actual quality is maintained of like whatever this material is at the top of the supply chain, it's actually like what it's supposed to be going into the product. And then from our side, like as a company owner, because if you guys don't know, Guerrilla Chemist has his own company too, and has a lot of, cool shit, and he knows the stuff when it comes to the chemistry side of things. Like what kind of falls on us after it goes through all of those phases? Like what do you expect to see at the top down to like the bottom?
Okay, so the top, when it comes to supply chain, they supply a C of A, and normally as a manufacturing facility, you have to qualify that vendor. So that means that sometimes you do a whole audit of them. Sometimes it's virtual, sometimes it's in person. You qualify their vendor, make sure that what they're sending you is what they say it is. You do your own verification, whether it's in-house or third-party stuff. Like for botanicals, you can do a simple TLC to say like, yes, this plant is green tea extract or something like that. If you're looking for something that's specific, a call out, like you look at the method on the C of A and it says, 50% saponins via I don't know, HPLC or UV vis or whatever, then you as the manufacturer have to make sure that the vendor does that. So you either do it yourself as a third party one time or in-house, or you qualify that vendor and use them enough to know that, yes, we've gone through the process, and there's forms and everything that you have to go through to qualify a vendor. And then you, as a contract manufacturing facility, if you get the material in and you deal with the vendor, then what your job is to make sure that the finished good has the raw materials in it and what they're supposed to be, right? So you develop your own SOPs for what you test for. A lot of companies specifically ask for certain call outs. So if I ask my manufacturer, I want to make a label claim saying this has 400 milligrams of caffeine or something, then they actually will run that test in-house, or if they can't do it in-house, they'll do it third party and verify for me that it does contain that. And that's usually done like HPLC or something like that. So, and you and I as the company owner relies on the manufacturer, who should be certified GMP, which means that they do everything to certain standards, and there's rules and guidelines for that, who also qualifies the vendor. So there shouldn't be stuff that trickles down into your and I products that's garbage, because at some point it should have been caught.
Right. So
does that make sense?
Yeah. So like once it gets down to a finished product, though, pulling a random bottle from a batch, like that would be something for like I don't know, like a hyper paranoid person would have to do that to make sure their shit is quality, right? Like you're kind of thinking the manufacturers have already established this.
Yep.
At the level of the supply chain at the top, they have this material that's been standardized to whatever, verified through HPLC, gets down to the manufacturer, they verify it is what it is, and it's supposed to be, the standardization and everything. They put it in, and then if you pull a random bottle yourself without your manufacturer knowing, you send it in for high sensitivity testing. That's like, how common is that anyways? Like, do a lot of companies do that? Because I think, I don't know, like it doesn't seem...
Not a lot, for sure. I know certain people did. John Meadows was a big one who did that a lot when he was with us and with Granite. But no, it's not common for you or I as the owners to pull random bottles of all of our products. Now that's kind of going over, like we're not required to do that, but I have done that. You have done that. That's like extreme due diligence. But we're also supposed to trust our CMP certified manufacturer that they did their due diligence in order to verify all these materials coming in are what they say they are.
Now, you mentioned John Meadows. He obviously, somebody who cared a lot about his consumers, his followers, people he imparted information on, great educational resource, rest in peace. What testing has he ever used ABC by chance? Like, I know a lot of people do trust ABC, and I feel like in this situation they've kind of got like a weird I don't know, like a connotation put on them in a bad rap simply because of a pharmaceutical error in the past. Like they've since rectified it presumably, or else they wouldn't be operational still, and they've come under new ownership. But like, I guess I'm just trying to bring to light how many people actually trust them currently, because it's not like they're known for doing stuff that a lot of people can't test for too, right?
Yeah. So I can't speak for John. I don't know what lab he used specifically, but I'll give you an example of when I used them, right? I had a sample made of a product, and this was years ago when you can use like 1,3-DMAA and DMHA, and I had this product made. Yeah. And it had both of them in there. And it was a high stem product. And obviously, like the more things you have in there, the more complicated it is. But through HPLC or UPLC, you can get good separation. And we tried the sample, and it was great. Really, really good. Really euphoric. I was like, okay, this is the one. And then when I actually ran the product and got the finished good and tried it, there's something was off. And so I wound up sending that sample that I got of the finished good, not the actual finished good, but the sample, the R&D good, to get tested off, and it had a peak on it, and this was through like LCMS, and the mass spec, which we'll get into how that works later, that the mass of that peak was the same mass as like an amphetamine based compound, very similar to Flocka, like off by like 1 atom to like that drug Flocka. I really am blanking on the research chemical of the compound, but you used to be able to buy it. And it was a very strong stimulant, like Adderall, way stronger. And so we found that peak in there, and it was just, wonder, well, why is this in here? But that was through ABC, and they bought the standards for 1,3 and 1,4 DMAA. They bought the standard for DMHA. So I never had an issue with them, and I know tons of people use them. They're not a small company by any means.
Yeah, and like they're going out to acquire these standards and whatnot. Like it's not cheap. Like just to get like a pharma grade version of a standard that's like 1.5 milligrams. It's like, you know, it could be like in the thousands, right?
Yes,
yeah.
It can be.
Yeah, so that's wild to hear about the stimulant thing. But getting into the actual like variability between different detection methods and like analyzation techniques. So this first link I sent you was a COA using HPLC for our turkesterone, and this is so Adriga turkestanica yielding 10% turkesterone, identity through TLC, and then assay of let's see, liquid chromatography for the actual concentration amount, showing it complies and is exceeding the 10% minimum by 0.73%. So that's sort of a thing that's like top of supply chain before hitting manufacturer. It's already being HPLC'd, right?
Yeah, that's your supplier, your vendor is supposed to be doing that on their batches, and that's how they generate the C of A with those specs. So when the person comes to you from that vendor and says, "Hey, I have a 10% extract for you. Here's the C of A. This is the method we use to validate it." That's how a facility would know. Okay, this if they're qualified, this is what their C of A said. It looks like it could be a good material. We test it ourselves. We see what it comes back as, and we use it.
Okay, and then it gets down to manufacturing. As you mentioned, they have their own quality control standards that you as a finished product marketer, or whatever you want to call, like the person who actually has the brand, you're trusting based on their certified, you know, they maintain their certification with their GMP standards. They're doing all of that stuff, and that's like assured via FDA regulation, right? To ensure it's actually okay.
And they get audited for GMP compliance too. So, you know, for manufacturing facilities, if they're not GMP, if they're not certified GMP, you don't want to use them. That's like the Wild Wild West kind of thing, you know.
How many people use non-GMP manufacturers? Is that like common? Because I didn't even, that seems like insanely risky.
I mean, all the good, and I don't want to say good as in like good product, but the people I know that I trust that have their own supplement companies, they all use certified GMP manufacturers. I don't know anybody who doesn't use one, or like not anymore. Like that used to be common like 8-10 years ago, but now things are a lot more strict.
So presumably if you wanted, like, I guess if you were going to sell like a research chemical or like DMAA now in a pre-workout, for example, I don't know, like certain smart drug nootropics or like whatever it is that are not DSHEA compliant have since been, you know, like banned by the FDA. Those are the kind of things, presumably, those individuals are going to a non-GMP manufacturer in order to get that done, or else, because no GMP manufacturer is going to touch that shit, right?
No, some of them, depending on what certifications they have, like if they have water or safe for sport, they can't even have that stuff in the building.
Damn.
Ample nothing. So they're that strict about that stuff. Yeah.
I think you cut out for a sec. You said NSF something.
Oh, and WADA. So like
okay,
the Olympics. So those type of regulations, like you can, if you have those certifications as a certified GMP manufacturer.
Okay. I hope people don't get annoyed at my lag time on Zoom, because I'm just noticing your video to audio ratio is like, hopefully I can fix it in post-production. Have you had this issue on podcasts before? I don't know if it's my internet or your internet. There's while you're talking, there's like a lag time.
Oh, I'm sorry, dude. No, my Wi-Fi is good right now. It should be working because obviously no one's on it but me. And I'm right by the Wi-Fi. But hopefully we can fix it. Can you hear me or no?
Yeah, I can hear you good. It's just the audio lag. Hopefully is it doing it on my end? Like when I'm talking, does it look like the words are coming out at the right time?
Yeah, it does. You look fine. So
Hmm. Okay,
I'm
well,
sorry.
apologize to the audience if it's like kind of weird. I don't know. It's probably my connection. Okay, so bring up the second link there. So we have this was a random bottle I pulled out of a batch. So the result is from last year, and this was using mass spec, so liquid chromatography with mass spec on top of it. So like from a accuracy standpoint or ability to stratify ecdysteroids from like the parent plant and all of its random constituents, like this would be something that's highly impossible to cross detect with like an ecdysterone, presumably.
So mass spec, the way that works is you still have the liquid chromatography part. Your sample is still enters as a liquid. There's still a UV detector lamp that shows the wavelength at which these molecules absorb light. And where it comes into play is the mass spec part, which breaks the molecule into different, like it has different cracking points. So it makes all these different ions. And when you get a readout, it's very, very more it's sensitive. So you can calculate the mass of these, that the parts of the molecule that have been broken up through like electrons that break them in. You can kind of see the difference between the mass of, turkesterone, which I think is like 494 or 496 grams per mole, versus like the ecdysteroids, which is like 464. And the difference is there's like a hydroxyl group on turkesterone that's not on one of the ecdysterone. So like the sensitivity is much greater. Like HPLC can separate stuff like called retention time, and it's based on polarity and that kind of thing. So you can get good separation, but mass spec is much more, has a lower limit of detection, and the sensitivity is much greater than just HPLC.
Okay, so this, presumably if you were going to assess something, this would be like gold standard for stratifying, like making sure it's there kind of thing.
Yeah. So for an example, in grad school, when I was in grad school and we were synthesizing stuff, in order for it to be published, and we had to go through LCMS, and we had to make sure that the mass of this is exactly weighed the mass of what we were supposed to make.
Oh, okay.
So
Okay.
yeah, very specific.
Awesome. All right. So at the top of the supply chain, you have the HPLC, which is, potentially good enough as is, and then you have at the manufacturing level, they're then verifying that, and then you have random bottle pull, which shows through LCMS it is what it is, and it's within striking distance of target. So for us, like I wanted at least 25 milligram yield for turkesterone per capsule, given the ratio of our cyclodextrin complex that we have in there. So like for us, this was like beyond passing by your interpretation, presumably. At least it was for me.
Yes.
Okay. Awesome. So I just wanted to make sure, because I'm sort of learning a little bit more about the chemistry. Like it's not like this is my exact forte of understanding like my subject matter expert area. So I just want to make sure there was no, due diligence being missed here. So I'm glad.
No, it's the LCMS, like I said, it's much more sensitive. It can separate the difference between those two compounds to a much better degree because it does it by mass versus just polarity that HPLC does. And then like the last one, like some people use UV vis, but that in my opinion, that's not a great one because a lot of molecules that are very similar in polarity like this and absorb light in the same region, it's very hard to separate them. Right. So UV vis would be the last end that I would check my stuff on if it were like something this close together. You'd have to do like LCMS or something similar to that.
Okay, so we have the third link there is another LCMS from 2022. So this is like a few months ago, or like, I don't know, 4 months ago. I don't know. It's like June right now, back in March. So like 3 months ago. So this is using LCMS as well and showing the ratio of our adjuga trichestenica relative to hydroxypropyl beta cyclodextrin and showing a conformation with LCMS And then we also, the link underneath that is a corroborating UV vis, which understandably, if that was on its own, it probably, I assume, would not be like certain. There's no cross detection in there. But when you see a corroborating LCMS with it for the exact same material from like a random bottle drawn, like to you, is that reassuring enough that it is like what it's supposed to be?
Yeah, I would, when you have the two together, even though like what I just said, HPLC is not the greatest, it can determine that you have this compound in there. It's just not the great at as great as quantifying it versus your LCMS. But since you've done both, and LCMS, like I said, is more sensitive and it kind of correlates, I think you're pretty safe to make a claim that there's this amount in each capsule.
Right. Okay. Like when you see chemistry analysis, like lab to lab, can it be variable based on like who the chemist is? Like I talked to another very, very credentialed chemist right before we got on this call, actually, and he was telling me about it's almost like art to some extent. Does that sound like somewhat accurate?
Yeah, every chemist has a specific way of doing things, and then there's always, you know, propagation of error, which the chemist should be making sure of. Like, as from the second you weigh the sample on the balance to the second you put it in the solvent to try and extract it to the second that you weigh it out to do the concentration and the molarity of the solution, to all of these different things have error. And yeah, it's kind of like an art form, the way you do certain things. And there are different techniques that most people do, the way you pipette. Like some of these instruments are extremely sensitive. So if you put in like 0.001 milliliters of something off, or 0.01 milliliters of something off, that could throw off your whole measurement, you know? So if someone's not doing something correctly, there can be. But assuming that they know what they're doing, then there shouldn't be a huge sway. You know, it would be close. And plus, you're comparing it to a known standard, right? So like all of these compounds that you're doing, you get the, you know, Sigma Aldrich standard and you run that first, and then their instrument knows, okay, this is exactly where the peak should be at this much concentration. And then you run yours, and based on the calibration curves, it gives you how much is in your specific product. So yeah, it is kind of an art, and it can be, you know, if the chemist does something incorrectly, it can throw it off. But it's not where it shouldn't be huge unless they really messed up.
Okay. And getting a standard like Chromadex and Sigma Aldrich, like these are like gold standard places, presumably, right? Like they're kind of like,
yeah.
Okay.
Those you recall like 99.9, 99.98, 99.8% purity. Like those are what you use to measure as the standard of caffeine or whatever you're trying to measure, right?
So who lab tests their stuff? Like themselves, or like how's that work?
They have to do everything they do, I believe, has to go through an ISO accredited lab to make sure that it meets spec.
Okay.
Because highly specific ways that they, you know, that they synthesize this stuff, and certain techniques for evaporating solvents that like to get it exactly a certain way, where like residual solvent can be on, let's say, a good that we get from a vendor, right? It could say plus or minus 1.1% residual solvent. Like these compounds are the standard. They can't have any of that stuff.
Right.
So I believe it, like an ISO accredited, which is like the top of the line laboratory, has to validate that. And it and I believe it goes by the NIST, the National Institute of Science, and I'm blanking on it. And but NIST is the one I think that everything has to go through.
Okay.
Sorry, I forgot the name of it.
Oh, no worries, dude. So when it comes to ISO accredited laboratories for dietary supplements testing, like third party, that's something that, like, I got into this industry maybe like
technology. Sorry.
Do you have a blip?
National Institute of Standards and Technology. Sorry.
It
was bugging me. I had to figure it out. So,
so
go ahead.
ABC, they are an ISO accredited lab that, like, have you ever assessed their, like you mentioned that one example where you had a spike of another compound and you kind of wanted to figure out what it was. And so presumably you've like audited their chemistry before and it all checked out.
I've seen multiple like chromatograms of different products from them, because I've used ABC to test for some of my current stuff with Chemix, as well as previous places I've used. And the manufacturers that I've used before have also used them. So I had a lab, basically like a whole folder of things I can just look at to make sure that they look legit and they know what they're doing. I personally have not gone to their facility to see them, but that's very rare that somebody does that, like an in-person audit of a third-party testing lab, unless you live in that state.
Right.
So that's not common practice.
Okay.
But they provide you with anything you want, especially since being ISO accredited, that they show you all of whatever they have, what their capabilities are. I felt comfortable using them, yes.
And as far as like patented material, so this is something I've come to learn is interesting, like the way they have their own methodologies for assessing the standardized contents of certain constituents of like, parent plants or whatever it is. And
Right.
if you test those at a different lab, is it ever possible if you use like an HPLC to end up with like a wildly different result? Like if you have a KSM 66 or like I don't know, like I'm trying to think of another patented material off the top of my head, like a nitrosigene or something like that. Is it possible to end up with like wildly different results than the patented company, or like what's the process of even getting that patented material approved?
So depending on what it is, the company that owns the patent ingredient should tell you how they test it and how they verify it is what it is. We were kind of talking about this off air, but I'll tell you the story. I won't mention the name of the ingredient, but there was a patented ingredient that I was trying to get a method for to test on HPLC to verify that it is what it is. And that company told me they are still in the process of developing a method for this compound. Keep in mind, this compound has been on the market, I don't know, since 2018, maybe 17, something like that. It's been around a long time. They still don't have a method. So they told me the way they do it is similar to what you said, is they test by parts. So if it's if the total mass of this is, let's for argument's sake, say it's 1,000 milligrams, and we know it's 40% of one compound and 30% of another compound and 30% of another compound, then you can just run the HPLC on those three things. And in theory, after, drying, it should come up to be that ratio should be correct. But that won't necessarily verify that product is, I don't know, like covalently bound or anything. Like for example, citrulline malate, there's no covalently bound citrulline malate. It's just citric acid poured in with like L-citrulline. So you would just be able to test those individually. You wouldn't see a chemically bound or ionically bound malate anion to citrulline.
Yeah. It seems it's interesting how many companies get away with that on their label too. The citrulline malates kind of like inflation thing. I don't know. I feel like people kind of know about it though now and kind of know how to.
They do, but I mean, even me, when I first came into industry, having a chemistry degree, I was under the impression that if you're going to label it citrulline malate, that's exactly what it is. But then I found out very, very quickly that is not the case with a lot of things.
No, I was surprised too. It's just like blended in a vat or whatever, and they just like put it in and say it's chemically bound. When in reality, it's because a 10-gram citrulline malate sounds a lot more impressive than like 6.6 citrulline and 3.3 malic acid or whatever, for sure.
Right.
Yeah,
exactly.
Cool. So I have a shit ton more questions, but I don't want to keep you too late. I think we could definitely revisit on like a long form podcast in the near future and discuss. I definitely want to talk about your fertility stuff and I don't know, just like novel stuff that you've been discovering in supplement industry and whatnot, and maybe get more into quality control and stuff. But I appreciate you coming on at such short notice. It's very much appreciated to help elucidate some of this information for the consumer who just like wants to know how the stuff works and, you know, how the supply chain functions and how their products end up the way they are. So I just want to say thank you for coming on.
You're welcome. Thanks for having me. And then, yeah, it really just comes about to trust. You gotta trust that your manufacturer trusts their vendor and that they've qualified them. And then you, as the person who's making the product, trusts your manufacturing facility. And then you, the consumer, trusts you or me, the person who's selling the product, that they did their due diligence. Unfortunately, we're in an industry that doesn't have as tight as regulation as it could. So by going above and beyond to show people like, yes, this is our products, this is what they contain, I went out and did this on my own. That shows a lot about, you know, character of the product versus, you know, companies that just push things out.
Right.
So
Awesome. Appreciate your time, man. Appreciate all the information and knowledge. And yeah, let's be in touch soon. I want to talk about the fertility stuff.
yeah, for sure. Let's circle back on that. I would love to.
All right.